Today, I'm going to present virtual screening as a strategy for finding new drug possibility specific for COVID-19. Even though there are several strategies, but I would like to focus on the virtual screening. Virtual screening is the computational technique or in silicon approach used in drug discovery to search library of small molecule in order to identify those structures that are most likely bind to the drug target, such as protein receptor or enzyme. So it requires key prerequisite knowledge about the species and energetic criteria responsible for binding of particular candidate ligand to receptor under investigations. It involves design of silicon filters to eliminate the compound with undesirable properties such as poor activity to select the most promising candidate. Its aim is to score, rank, or filter a set of structures by using one or more computational procedures. For hit identification, it involves pre-screening filtering to filter away the compound with unwanted properties. Next is the virtual screening, which involves preparations of database structures, for example, by changing 2D to 3D. Next is database screening, for example, molecular docking and scoring. The last one is the post-screening filtering, which involves hit list reductions by QSA. The functions of VX basically it is used to decide which compound to screen and which libraries to synthesis to enrich the library of available compound. It is used to decide which compound to be purchased from alternate company, and it also used to analyze the result of experiments such as HTS run. For VX, in an effort to show that searching for lead candidate by using computer is a serious alternative to HTS. Unlike HTS, which is an experimental approach and requires considerable resources, VX is a theoretical approach utilizing various computational methodology to screen compound collections and virtual compound libraries. It gives higher success rate, clear structural hypotheses, and improved understanding of drug-receptor interactions and molecular recognitions. Besides, it reduces the exchange cost and human resources spending. Importantly, since there is overwhelming demand for COVID-19 drug, it helps to save time for drug development. Virtual screening consists of two methods, ligand-based and structure-based. The first method, structure-based, it is a computational approach by utilizing the known 3D structure of biological target obtained from the NMR and X-ray crystallography. It is used to dope a collection of chemical compound into the binding site and select a subset of compound based on the predicted binding score for further biological evaluations. It involves molecular docking and de novo design, which we'll explain later, and it also involves the pharmacophore modeling. However, there is a difference of pharmacophore modeling between the structure-based and ligand-based. For structure-based, it is generated from the receptor binding site, while for the ligand-based, it is from the bioactive conformations of ligand. Last one is the MD, which is used to analyze the physical movement of atom and molecule. For molecular docking, it is a method that predicts the preferred orientations of one molecule to a second when bound to each other to form a stable complex. Then the complex quality is evaluated by score to detect the correct bond conformations and estimate binding affinities. Next is de novo drug design. It is an interactive process in which the 3D structures receptor is used to design the newer molecule. The next method is the ligand-based. It is the method that relies on the knowledge of other molecules that bind to the target of interest. Because the 3D structures of biological targets is unknown. It involves three components. The first one is a similarity search. It is a method for screening database to identify the molecule that are most similar to the defined reference structure. For the next one is the pharmacophore model. It is a set of points in space defining the binding of ligand to receptor. So the candidate ligand will be compared with the pharmacophore model to determine the compatibility. And the next one is the QSAR. It is a measurement of predictable variables to behavior of response variable to predict the activity of new analogs. Lastly, I would like to share the latest research about the drug discovery of COVID-19 by using the computational method by Wu in 2020. It involved the screening of zinc drug database and natural product. Besides, the database of used antiviral drug is screened by using the virtual ligand screening. So the result shows that there are 4 types of drug target for COVID-19. The first one is the target inhibiting virus RNA synthesis, target inhibiting virus structural protein, target inhibiting virus factor, and target blocking host specific receptor or enzyme. This is the reference. That's all for me. Thank you.